Bismillahirrahmanirrahim. Assalamu alaikum. I'm Dr. Ismana Shahid and I'm working as Assistant Professor of Community Medicine at Rawalpindi Medical University. Today I have to discuss some of the basic concepts related to biostatistics. By the end of today's brief session, I hope listeners will be able to define biostatistics, illustrate the importance of statistics in medical sciences, and discuss descriptive and inferential statistics. Biostatistics, as the name indicates, it includes the biostatistics, as the name indicates, it deals with the whole process of collecting, summarizing, organizing, interpreting, presenting the data related to medical sciences. So, statistics is basically the process that starts following data, that also includes the process of data collection till its presentation or its publication in various journals for dissemination of data to the readers. There are two main types of statistics. It is very important to understand these two statistical terms in order to make their use while composing the manuscript or compiling the results. Descriptive and inferential statistics. Descriptive, as the name indicates, is meant to describe the data. So whatsoever data has been collected, in order to describe the data, we make use of measures of central tendency, measures of dispersion, and drawing various tables, charts, and graphs in order to present the data. Measures of central tendency include mean, median, and mode, and measure of dispersion includes calculation of standard deviation, range, interquartile range, and variance, etc. Whenever we calculate mean, along with mean we calculate standard deviation. Calculate median, along with median we calculate interquartile range. We also draw various tables, charts, and graphs in order to get the attention of readers, because tables, charts, and graphs present the data that is very conspicuous and easily understandable by the readers. So descriptive is meant to describe the data, whatsoever has been collected. And in order to describe the data, we calculate various measures of central tendency, measures of dispersion, in addition to drawing various tables, charts, and graphs, etc. So whenever we apply descriptives in terms of calculation of measure of central tendency or dispersion, or making use of various graphs and charts, then we mention the word descriptive statistics were applied in our manuscripts. Just mentioning that descriptive statistics were applied shows the use of all these measures. Inferential statistics, as the name indicates, inferential is meant to conclude something, to reach a conclusion, to bring out the inference. And in order to scientifically and statistically prove the results, we make use of inferential statistics. Descriptive, as I have already discussed, is meant only to describe the data. Descriptive we are meant only to present the data, describe the data, whatsoever has been collected, what was the mean age of the participants, how many males or females were included in the study. So just to describe whatsoever data has been collected. But in order to reach some scientific conclusion, to scientifically, statistically verify our results, we make use of inferential statistics. Inferential में हम टेस्ट ऑफ सिग्निफिकेंस अप्लाई करते हैं, एंड अल्टीमेटली पी वैल्यू की थ्रू वी verify our results, confirm our results. So inferential statistics includes two main categories. It includes the calculation of confidence interval, and it includes the calculation of p-value following application of various tests of significance in order to measure the association of two variables or more than two variables. For example, you can see in this slide that mean BP of Americans by applying test of significance confidence interval from a single sample was found to be whatsoever data has been collected. So in order to compare the mean blood pressure of two groups or more than two groups, we apply the suitable test of significance, and following application of that test of significance, we see whether the manually calculated results were significant or not, whether the manually calculated differences among two or three groups were significant or not. And depending on the p-value, then we conclude and we accept or reject the null hypothesis. Hypothesis is basically a supposition that was drafted or that was given before the application of test of significance. Confidence interval is basically the claiming something confidently about the population, because we are unable to calculate the collect the data from the whole population, so we make use of sample. चूँकि पॉपुलेशन से पूरी पॉपुलेशन को स्टडी में डिफरेंट कंस्ट्रेंट रिसोर्स की वजह से या किसी और लिमिटेशंस की वजह से पूरी पॉपुलेशन को स्टडी में इंक्लूड नहीं किया जा सकता. दैट्स वाई वी select a sample from the population, and depending on the values or characteristics of population,